Okay, time now for the weekly arithmetic moment. What does it take to build one of these? This is the point of this is not simply to go through some exercise with numbers, but the fact is the numbers often illustrate the scale of thing that the cell is trying to accomplish and how that scale constrains what cells can do. So let's go back to 8 nanometers per tubulin dimer within a microtubule lattice. And that means there's 125 tubulins per micron of protofilament. The doublet microtubule that makes up that's characteristic of the axoneme, there's 13 in the A tubule and 11 in the B tubule. That's the partial one over here. So that means 24 times 125 per micron. That's going to be 3,000. Okay, there's 9 doublets. Ooh, but there's also a central pair. So let's just say approximately times 10, that's 30,000, right? 30,000 tubulins per micron of ciliary axoneme. On a starfish larva, the cilia are probably 25 microns long. So times 25 microns equals about 750,000 tubulins, just the tubulin alone, not to mention the dyneins, of course. 750,000 tubulins for the axoneme. But that's not all. Cilia actually won't beat well without these roots holding them in place. In fact, if you drop a sea urchin larva in a microtubule poison, the microtubules of the axoneme are relatively stable. They won't fall apart, at least not for a while. But a lot of these other microtubules in here will, and those larvae can't swim in microtubule poisons, largely because the ciliary beat is completely discoordinated. So it turns out for the microtubular roots in a typical epithelial cell, you've got roughly twice the microtubules as in the axoneme. Who knows if you need all of them, but at any rate, it takes something like, the bottom line is it takes something like, let's say, 2 million tubulins, plus all the tectins, plus all the dyneins, etc., etc. The tubulins are the most abundant proteins, of course, to make each cilium in a starfish larva. All right. Now, recall from a particular gene, you might be able to make 1 mRNA per second. So accumulate with no turnover. It takes you about 20 minutes to make 1,000 mRNA, let's say. Once you've got those 1,000 mRNAs, you've got to translate them into proteins. Roughly, it'll take you about, since it's, let's say, 1 protein per mRNA per second, it'll take you roughly the same amount of time to accumulate 1 million of them. You can see where this is going. You could probably make all the stuff that you need for a cilium in about 30 minutes to make the proteins in a cilium. That is, if you didn't have them handy already. And many of these things do require new gene expression. In fact, the tectins that link these doublets together are one of the earliest genes expressed in the sea urchin blastula, as are the tubulin genes and dynein, unsurprisingly. Experimentally, you can knock off all of the cilia in a sea urchin or a starfish embryo. You can put them in hypertonic seawater, about 50% more salt than usual, and all the cilia will fall off. And it takes them about 30 minutes to grow them back. If you knock them all off and you put them in protein synthesis inhibitor, it takes them about 30 minutes to grow them all back, but you can't do it again. So they do have enough stuff in the cell, besides the stuff in the axoneme, that they can build another cilium, and it takes about that time scale just to assemble this structure, even if you can't translate new proteins. But to be able to consecutively make replacement cilia, they have to have new protein synthesis to make all that tubulin and dynein and so on. Now, ciliary length is highly variable from cell type to cell type, but it's very consistent amongst cells of a given type. Around the blastula of a starfish, there are a few exceptional ones, some non-beating cilia up at the anterior end, for example. And many embryos feature extensive ciliary differentiation, as do protists and so on. But from cell to cell of a given cell type, ciliary length is fairly consistent. Let's swing over to the other side of the board to consider a classic experiment on the green alga Chlamydomonas, which is a favorite for studies of ciliation. Chlamydomonas is as close to a spherical flagellate as you can get. And it has two flagella that are typically about as long as the diameter of the cell. That is about 10 microns, maybe a little more. And Chlamydomonas swims with breaststroke most of the time when it swims forward. The cilia both have the same kind of power stroke and recovery waveform. When it undergoes a reversal, it actually beats these cilia with a flagellar waveform instead and swims backward, but we'll get to that later. So the experiment is as follows. We're going to sever one of those cilia, knock it off, just one of them. Ouch. Now, in the old days, nowadays you'd do this with a laser or something like that. People like lasers much better. In the old days, they would do it with a little tiny torture chamber that squeezed the cells or something like that. Or there are a variety of chemical treatments that will knock cilia off, and you can just look for the cells that have lost only one of their cilia. Many of these experiments were done in paralyzed mutants of Chlamydomonas, so that they will hold still on the microscope the whole time. So immediately after severing, this is the circumstance. Within a few minutes, the severed cilium, and just to confuse everybody, Chlamydomonas people call them flagella, not cilia. The severed cilium has started to grow back, while the intact one has begun to shorten. 20 minutes. The severed cilium, I'm going to insistently try to call them cilia instead of flagella. The severed one has regrown to about half its original length, and the intact one has shrunk to about half its original length. Takes about 40 minutes for Chlamydomonas to mostly regrow its flagella. And the time course looks like this. So if this is the standard 12 micron length, one of them, the intact one, starts out there, the other one starts out at 0, and I don't know, maybe there's a little lag. It grows steadily until it sort of plateaus like that, whereas the intact one is shrinking steadily before regrowing also. Let's say the whole time course is 60 minutes. So how do you interpret that? Why does the intact one shorten? Surely just to be friendly. Okay, there's always a stupid why and a not so stupid why. And then there's the teleological why, okay? That is, what's the purpose of doing it this way? Well, one point to make is that it behooves these cells, if they want to swim straight, to have two equal flagella. So if it's going to take you a long time to restore a full-length cilium, if it's going to take you a long time to restore the full-length organ, then maybe shortening the other one means that, okay, maybe you can't swim as fast, but at least you're not swimming in circles. Okay, mechanistically, one possibility for what's going on here is that the cell is cannibalizing the intact organelle in order to rebuild the severed one while waiting for newly made parts to accumulate. That's a realistic possibility. It happens to vary from strain to strain how much of regrowth you get if you prevent protein synthesis. So if you block protein synthesis and knock both cilia off, Chlamydomonas, and again, this is the 12 micron standard size here. If you knock both flagella off, you get regrowth to about half their normal length, suggesting that in the absence of protein synthesis, in the absence of new gene expression, the cell only harbors about enough to make one cilium, one standard length cilium. Of course, so that's in cyclohexamide over here. That is no new proteins. Normally, the response would be for, if they weren't in cyclohexamide, they'd rebuild them entirely within an hour. Now, the normal response for wild type cells, if you knock off just one of them, is what I drew already. And that's what will happen in cyclohexamide as well. As I mentioned, many of these experiments were done in a paralyzed mutant, so they could watch what was happening as it sat on a microscope slide. For whatever reason, these paralyzed cells don't have as much of a reservoir of precursor. So if you knock both of the cilia off in cyclohexamide, you only get a couple microns of ciliary regrowth. And in the paralyzed mutant, If you knock one of them off, again, here's the benchmark. If you knock one of them off, you get regrowth to a little more than half. The suggested interpretation here, again, is that the cell harbors some reservoir, which in the case of this particular mutant is less than the wild type cells. The cell harbors some reservoir of building blocks, and it needs to cannibalize the existing organelle in order to get a jump on rebuilding the missing one. How does it know? How does the cell know when these organelles are the same size? Size equalization of the cilia in these cells and many others turns out to be explained by a rate-limited assembly process that builds the cilia at their tips. The axoneme is being built continually at its tip and also disassembled continually. In the regeneration process, forward transport is prioritized to the damaged cilium so that this one is starved, the intact one is starved of precursors that it would need to maintain its length. And so it begins to shrink. Once lengths are equalized, the forward and so-called retrograde transport rates of building blocks are also equalized, and the two cilia grow at the same rate. This is an early hint at the existence of a process that was only discovered much later, and we'll read a paper about the discovery of intraflagellar transport. Like many of my favorite discoveries in biology, this one was made through what a friend of mine likes to call LCPA analysis. This stands for look closely and pay attention. This fellow, Keith Kosminski, was looking very carefully at his Chlamydomonas cells, and he saw little wads of stuff tracking up and down the cilium as it was spread out on a cover slip beforehand. So looking in here at the cilium, let's sketch in the axonemal microtubules. What he visualized with careful adjustment of his microscope was some little blobs of stuff, seemingly in chains, ganged together, moving at a steady rate up and down the axoneme. Whatever he saw, if it was moving this way, it kept moving that way. And if it was moving this way, down toward the base of the cilium, it kept moving that way. These trains were slightly distinctive in a few other ways. It turned out, ultimately, that the retrograde transport, the trains moving that way, are moving because they are driven by cytoplasmic, not ciliary, by cytoplasmic dynein carrying these trains toward the minus end of every microtubule, which remember is down here at the basal body. And I guess I need another color for it, but the so-called anterograde trains, that is the ones moving out toward the tip, are moved by a member of the kinesin family of molecular motors. Actually, I can't remember whether it's the second or third kinesin ever to be discovered, but it's called the kinesin 3, or the intraflagellar transport kinesin. This intraflagellar transport mechanism is almost universal. Anywhere that people have looked, that's how the eukaryotic cilium is built, and it is the basis for many of the adaptations of the cilium to make things like the photoreceptors in our eyes, which are really highly modified cilia. Here's a question that's worth a moment's thought. If dynein is bringing things back from the tip of the cilium, that is, it's taking apart the axoneme at its tip continually, while kinesin-driven trains rebuild them so that the tip is always in flux from these two activities. How come drugs that poison cytoplasmic dynein cause cilia to shrink? There are surprisingly few ways, actually, to treat cells to stop their cilia from beating or prevent them from growing cilia, but one of the effective drugs happens to target the cytoplasmic dynein that drives retrograde transport. So how come blocking retrograde transport makes the whole thing shrink? Well, obviously, because once these things get out, if there's nobody to take them back, they'll accumulate out here, and nobody will be delivering stuff anymore, right? So blocking transport in either direction stops the whole process and leads to the failure to maintain the axoneme. So finally, let's review. We've got cells like a garden variety epithelial cell that has a simple cilium that it beats with a standard power stroke and recovery. You can gang a whole lot of those together if you want to move more fluid, or you could make multiciliated cells that express many cilia on their surfaces. We've talked about things like sperm that have long cilia and use a flagellar waveform. These are basically screwing their way through the water. I'll draw it approximately size equivalent. We've got things like this flagellate chlamydomonas that has two cilia that it beats with this kind of standard power and recovery stroke to breaststroke through the water. And of course, we've also heard about things like modified cilia, the cone cell, for example. Various other instances. Beyond this, one of the common strategies to make bigger, more powerful swimming organs is to make compound cilia. Make a lot of cilia, but don't just wave them around independently. Many mollusks make a great big blade that consists of many cilia lashed together, they can make a longer cilium this way that retains the power and recovery stroke, has greater hydrodynamic reach. But there's one little puzzle based on the arithmetic exercise that I went through, which is especially pronounced in early embryos, for example, of mollusks and annelids. Some of you may have seen trochophore larvae in some other contexts, which swim not by giving every cell a cilium, but by making a band of a few cells whose only job is to make cilia. And these cilia are big, long, compound cilia, like these, that beat much more strongly and propel these much faster than any sea urchin or starfish larva goes. So if the arithmetic exercise that I went through is meaningful at all, how could they possibly do that, especially in an early embryo? I've made this argument that the great rush from the zygote to blastula is all about having enough DNA that you can manage to actually do something with your own genome, and I've emphasized the metabolic burden of being able to even make enough tubulin to build a cilium. How can these cells early in the embryo, at something like the 128 cell stage, manage to conduct all the biosynthesis needed to make 20, 30, 40 axonemes? One of the virtues of these kinds of arithmetic arguments is that if they appear to be proving that an everyday thing is impossible based on what you know about how it works, then probably you don't know something about how it works. At least one part of the answer for at least some species of mollusk is that they don't have just two tubulin genes in there to work from. Limpets, at least one species of limpet, is known to have about 24 tubulin genes in its genome, presumably to support the production of heavily multiciliated cells that have to make those cilia rapidly during development.